you. Thank you very much, sir.
Sanji, please proceed.
Yes, sir. Yes, sir. A very good morning, respected director, sir, faculty members, dear students, and audience. I, Dr. Sant Kumar Verma, Associate Professor, ISF College of Pharmacy, Moga, would like to welcome Professor Vinod D. Rangari, M. Pharm PhD, Dept, a renowned professor from Guru Ghasidas Central University, Bilaspur, on the online platform of ISF College of Pharmacy, Moga, for delivering talk on old molecules, new potentials, novel tool, and revolutions concept in drug development. So, sir, welcome you on online platform of ISF College of Pharmacy for 101 lecture series. I request to respected director, sir, please welcome our today's speaker and enlighten about the 101 lecture series. Over to director, sir.
So, thank you very much, Dr. Sant Kumar Verma ji. On behalf of ISF Dialogue Series and ISF family, first of all, I welcome Dr. Vinod Rangari ji from core of my heart, sir. So, I know that I met Dr. Rangari ji two times at a different platform, and I have also met in the one place. And I come to know that Dr. Rangari ji is a very dedicated person and very experienced in the research in the specific zone. So this is the good opportunity to all the youngest researcher to listen the lecture very carefully and take benefit. And I am also thankful to Dr. Rangari ji for accepting our invitation, and you have the accept for deliver a lecture in this 101 ISF dialogue series. So without delay, I know that this is the very compact system of the dialogue series, so that most of the pupils they will take the benefit of this series. So thank you very much, sir. And again, I welcome you. So over to Dr. Sanji. Welcome, sir.
Thank you, director. Now I would invite Professor Dean and Head, renowned scientist, Professor V.D. Rangari, sir, for his talk. Over to respected Rangari, sir.
आवाज डिस्टर्ब होऊन जातो. बघ आता ते येत नाही माझं तिकडलं. I hope my presentation is visible to everybody.
Sir, not right now. So please do. Yes, now okay.
Yes, it is visible, sir.
Yeah. So I think we should start now.
Yes, sir. Yes, sir. Yes, sir.
Okay, a very good morning to all the students, faculty members of IFS College of Pharmacy, Moga, Punjab. It's a great privilege for me to be a part of this 101 lecture series of your institution. As informed by Professor Sharma sir, Gupta sir, we are really good friends which met quite often during the seminars and conferences, and it was a privilege for me that after this meeting only we have gone into MOU signing with your institution, and through that the type of interaction and the collaboration has started with IFS College of Pharmacy. It's a matter of great rejoice for me, and I congratulate IFS College of Pharmacy management, principal, director, as well as faculty members and students for getting 24th position in NIRF ranking in 2023. This specific lecture is my continuation of the legacy of my PhD guide of Regional Research Laboratory Jammu, Professor Dr. C.K. Atal, renowned multidisciplinary scientist and the pharmacognosist and the ex-director of the Regional Research Laboratory Jammu, which is now known as Indian Institute of Integrated Medicine. There are reasons for dedicating this lecture to Professor C.K. Atal, because this is one of the best research that has been done by Indian scientists in Regional Research Laboratory, CSIR Laboratory, Jammu, and which I have continued during my academic and research programs. So with this basic information, now let's go to this lecture, Old Molecules and New Potentials, Novel Tools and Revolutionary Concept in Drug Development, vis-a-vis Rediscovering Drug Doses and Economics of the Treatment. For coining this type of title for this lecture is important. In three ways it is important: that old molecules and having the new potentials which have been explored, how it is helpful for the drug development and deliveries as a novel tool, and how by doing so we can rediscover drug doses and economics of the treatment. So all these three aspects we are trying to see in this lecture. As a student of pharmacy and the professional of the pharmacy, we know that for the drug development, there are a number of technologies we are developing, we have developed in the whole world, and the recent developments in the drug bioavailability enhancing technologies, revolutionary shift has taken place in the way medicines are administered. And in this administration, mostly the emphasis is to reduce the dose of the drug as well as the cost of the treatment also, and to some extent even the side effects of the drugs. We are all aware that it is the bioavailability which is the most important aspect of drug delivery, because in the minimum dose we have to have the maximum activity of the drug, which is generally prescribed to the patients or given to the patients by different routes of administration. So it is a measure of the rate and extent of the therapeutically active drug that reaches the systemic circulation and is available at the site of action in the body. Mostly intravenous drugs are considered as 100% bioavailability giving drugs, but generally orally administered drugs decreases the bioavailability because of so many different types of factors which we are going to see. We are aware that after giving the drug by the oral routes, when it goes to our GI tract, mostly GI tract, it is dissolved, it is absorbed, and through the first pass effects, it is generally brought to the systemic transformation. But while bringing it to the systemic absorption, many of the phases through which the drug has to pass, and because of which the drug is metabolized and cannot reach the systemic circulation in a sufficient amount. So these rate limiting steps of the bioavailability are mostly type of dosage form, the particle size, molecules in the solutions, dispersion, disintegration, disaggregation, dissolution, permeation, and many other steps are there for the bioavailability development. We can see that the number of our biological systems involved in this bioavailability affecting factors. Mostly the first is stomach. Mostly stability and solubility of the drug is mostly affecting factors in the stomach because of the pH of the stomach. Mostly when it goes to the intestine, especially in the small intestine, passive and active transfers play an important role, as well as the PGP-FX and the cytochrome 3A4 are also the enzymes which are responsible for the degradation of the drug. And when it reaches to liver, mostly large number of the cytochrome P enzymes which are present in liver mostly degrade the drug, because of which, or metabolize the drug, because of which it cannot reach the systemic circulation. So here is the concept of bioenhancers or bioavailability enhancers that we are putting. It is not a new concept, because I would come to the facts that how it is not a new concept. It is a very old concept of bioenhancer. So we can define bioenhancer as an agent capable of enhancing bioavailability and bioefficacy of specific drugs with which it is combined without any typical pharmacological activity of its own as the dose which is utilized of this. And this bioavailability enhancement may take place by different types of approaches, mostly permeability enhancing dosage form, which generally we do through liposomes and emulsions, prodrugs, particle size reduction, P-glycoprotein inhibitors for improving the bioavailability and drug delivery, and use of protease inhibitors, which are generally effective for degrading the cytochrome P enzymes. There are several types of compounds which are found in nature that are having the specific absorption enhancing effects, like bile salts in our body, salicylates, non-ionic surfactants, fatty acids, ionic surfactants, chelating agents, swelling polymers. These are some of the agents in the body. Along with that, other approaches were prodrugs, particle size reduction, and glycoprotein we have already seen. But the natural products which are mostly utilized for bioavailability enhancing, we have seen that several types of natural products which are generally antioxidants, they are bioavailability enhancers, mostly flavonoids. So type of flavonoids like quercetin, naringin, genistein, few of these important bioenhancers I would like to quote. Glycyrrhizin, which is triterpenic acid, triterpenoid glycosides, and nitrile glycoside, which is found in nature. Cumin extract is a very good bioavailability enhancer. Cyanomenin, piperine. These are some of the natural bioavailability enhancers which are mostly produced by the nature in specific medicinal plants. Now let's come why I am dedicating this topic, this lecture, to Professor Dr. C.K. Atal, Director, Regional Research Laboratory, which is now known as Indian Institute of Integrated Medicine, which has done and is doing wonderful work in drug development in the CSIR laboratory of Regional Research Laboratory, Jammu. Dr. C.K. Atal had been a director of this institution since about 1970s to 1985-86. And during that period of time, he has developed that institutions with a goal to utilize the natural resources for the drug development. And this is one of the best research output of Regional Research Laboratories, CSIR Jammu, which has been done by industrial collaboration with many of the research institutions, but basically, importantly, with Cadila Laboratories at RRL Jammu. And this Cadila Laboratories has achieved success in increasing the bioavailability of some important drugs by using piperine. Now, how the type of research work which has been done at RRL Jammu was very much trend-setting and associated with old molecule, new potential. I would like to take you to some of the age-old traditional systems of medicines of our country. Mostly, Trikatu is one of the important formulations in our Ayurveda and Siddha system of medicine. And this Trikatu is the mixture of three components: black pepper, long pepper, and ginger. All three are the spices. And this trigger 2 is the type of formulation. In fact, it is not a formulation, but it is a mixture of these three, which is generally utilized along with many different types of medicinal plants and prescribed in Ayurvedic and Siddha system of medicines. So, when in Ayurvedic and Siddha system of medicines, Dr. C.K. Atal and many other scientists who explored that why trigger 2 is being added in Siddha system of medicine with almost all types of medicines. So they have taken this project for exploring what is the use of trigger 2 along with the medicinal plants when it is given in the formulations. And with that, mostly they explore that it is a type of formulation which is having the large number of bioavailability enhancers, because of which it enhances the bioavailability of the medicinal plants, extracts, as well as the pure components. So now this trigger 2 which is there is the mixture of three: Piper nigrum, Piper longum, and Gingiber officinalis. Out of these three, Piper nigrum and Piper longum, which are long pepper and black pepper, they are the type of plants in which the major component is the alkaloid piperine. And this alkaloid piperine, which is present in Piper longum and Piper nigrum, is found to be a very good bioavailability enhancer. And this research was first of all initiated in Regional Research Laboratory, Jammu, with the initiative of Professors Dr. C.K. Atal. And the first research paper, which was published in 1979-80s, which was associated with the bioavailability enhancement activity of piperine with different types of therapeutically active component. It was such an important research which was taken cognizance by WHO, World Health Organization, that such type of molecule is there in nature, which does not show its own activity, but generally is involved in taking the therapeutically active molecule to the site of action without breakdown or without any type of degradation. So this was a wonderful research which was initiated and taken note by World Health Organization. And after that, in the whole world, the research was started. So this is black pepper or long pepper, which is known as Kali Mirch, which is obtained from dried unripe fruits cultivated mostly in South India. 5 to 9% of the pure, about 99 to 100% piperine can be isolated with the simple process. It is antioxidant, bioavailability enhancer, CNS depressant, hepatoprotective, and antipyretic and analgesic agent. So this piperine compound, which is obtained by a very simple process with alcoholic extraction and after that precipitation and crystallization with alcohol or ether, becomes an important commodity now for the drug development and drug delivery as a bioavailability enhancer. I would like to take you to the type of drugs and the mechanism of action of piperine. Mostly almost all types of categories of the drugs have been influenced by the piperine. Cardiovascular drug, respiratory drugs, CNS, GIT, antibiotic, anti-cancer, and anti-HIV drugs. But mostly, it has wonderfully worked in anti-cancer drugs and anti-HIV drugs. There are almost all types of compounds, all type of therapeutically active agents, right from antibiotics to that of anti-HIV drugs, which has been studied with piperine, and many of the industries have already filed the patent by protecting their rights with piperine. But up till now, they have not initiated the use of piperine as a bioavailability enhancer in their drug. It was only one company which was collaborating with Regional Research Laboratory, Jammu, that is Cadila Laboratories Limited, that only became successful, and that success story also I would like to share with you. So how it influences, mostly piperine influence the absorption of nutrients like vitamin C, vitamin A, vitamin B6, carotene, coenzymes, and selenium, and many of the other micronutrients. You would be aware that whenever we take the soup or the fruit juice, It's a practice in our country to sprinkle some of the black pepper over these juices, whether it is lime or orange juice or whether it is any type of soup that we do. Why we are doing it? It is because this piperine which is present in black pepper powder, even in very small quantity if it is there, it absorbs, it causes the improvement in absorption of these nutrients, because of which we get the better health benefits. It can also help combat the malnutrition because of these malabsorption states and chronic ailments. And dose of the piperine which is utilized as this bioenhancer is mostly 7.5 to 10 milligram in the all formulation, irrespective of the dose of the active drug present in this. How the piperine, which is an alkaloid, mostly acts as a bioenhancer. Mostly there are several mechanisms, and it is not just one mechanism by which it acts. There are several mechanisms by which it acts, and as we are doing more and more research on the mechanism of actions, we are coming to know many different types of mechanisms of the bioenhancer piperine. It works through DNA receptor binding, modulation of cell signal transduction, inhibition of the drug effects pump, PGP2 and P-glycoprotein 2 pump, production of glucuronic acid in intestine with which allows more active form of drug to enter body. It may increase the absorption of the drug in gastrointestinal tract and inhibits enzymes responsible for drug metabolism, especially in liver when the drug passes through the liver after absorption from GIT. So the oral administration of piperine mostly inhibits the UDP gluconoryl transferase activities, and mostly this type of activities are generally demonstrated by different types of biological studies. Piperine inhibits human P-glycoprotein and cytochrome P3A4, but along with that, some of the other metabolizing enzymes which are inhibited or induced by piperine include cytochrome P1A1, 1B1, 1B2, 2E1, 3A4, and many others. In general, it inhibits the drug metabolizing enzymes, stimulates the absorption by stimulating gut amino acid transporters of the drugs. This shows that the type of microsomes, cytochrome 450 enzymes and UDP-GT, which is mostly inhibited by piperine, are generally induced by the type of anti-cancer drugs, anti-HIV drugs. Most of the anti-HIV drug, anti-cancer drugs, they induce the cytochrome P450 and other enzymes, and because of which the metabolism of these active ingredients takes place in the liver, and because of which the bioavailability could not reach to the maximum level. So such type of enzymes are mostly inhibited by piperine. Mostly it is having the type of activity in the humans, but even in the experimental animals, it has shown that mostly in almost all conditions, it increases the bioavailability. Some of the components like vaccine from Vasica extract, propranolol and theophylline, aflatoxins, accumulation, serum concentration of curcumin, plasma level of coenzyme Q10, delayed elimination of phenytoins, amoxicillin, epigallocatechin gallate, which is the mostly types of compounds in the tea leaves which are good catechins for the body, antioxidants. All they have been increased by the piperine. And while doing so, 7.5 to 10 milligram of piperine, it reduces the cost of the drug, reduces the dose of the drug, increases the efficacy of the drug, and reduces the toxicity of the drug. So that is why it is associated with the economics of the treatments also. Now, this is one of the trend-setting example of the Indian research, that is from Cadila Laboratories. When this type of research was done and the papers were started getting published from Regional Research Laboratory, Jammu, that time Cadila Laboratories signed an MOU with Regional Research Laboratory with the first drug, that is the anti-TB drugs. We are aware that the TB is such type of chronic disorder drug, chronic disease, for which long treatment of rifampicin is required. And rifampicin is having many type of side effects, because of which sometimes the patient has to have the very poor health conditions during the long period of time. So this is semi-synthetic antibiotics, and mostly it is having the solubility very less in water. Its ED50 is LD50 is very high, and therapeutic dose is 450 milligram, and the therapeutic category is the antibacterial and tuberculostatic agents. So this molecule was taken along with piperine to study the bioavailability effect of piperine. I would not go into the details of the research, but this is a type of figure which indicates a lot. Mostly the blue line indicates, graph line indicates that it is the conventional dose, which is if reduced to 200 mg, how the bioavailability is reduced. And conventional dose, which is generally 450 milligram, what is the bioavailability of the drug in the green line. But as soon as the 200 milligram of the drug of rifampicin, if mixed with the piperine 10 milligram, how the bioavailability of the rifampicin shoots, and even better than that of the conventional 450 milligram of dose. So because of this research, the dose of the drug of rifampicin from 450 milligram was reduced to 200 milligram just by adding 10 milligram of the piperine bioavailability enhancer. So when the anti-TB drug risorin 10 milligram piperine and 200 milligram rifampicin, when this drug was launched in New Delhi, that time I was really a witness for that, along with Professor Dr. C.K. Atal sir, who was invited for this program, although he was retired and not in very good health conditions. So it was really a very important fact that the drug 200 milligram of rifampicin came into market by just adding 10 milligram of piperine, and it has changed mostly the drug development and drug delivery system very much for the other types of drugs also. With such type of initiative from Aral Jammu, when after my PhD from Jammu I came in my academic line, so I started working on this area. At least one M.Pharm or PhD student used to work, and with this motivated research base, we have started and worked on many different types of therapeutic agents for increasing the bioavailability. I would like to quote only two examples out of this motivated research. One is study of anti-cancer drug Taxol, and the another is the anti-HIV drug. Mostly Taxol, paclitaxel, we know that it is one of the very important anti-cancer drug, mostly produced from Taxus brevifolia. The plant is available in a very less area of the Arctic region, and the type of compound which is present in the bark of this drug is very less, because of which the drug is not available in large quantities. Semi-synthesis or the total synthesis of Taxol is not yet a successful one, and even the different types of things by developing the paclitaxel by plant tissue culture method had also not been successful. This drug, which is known as paclitaxel, is highly lipophilic drug, insoluble in water, and this is drug which is mostly metabolized by cytochrome P2C8 and cytochrome P3A4. So these enzymes play a very important role, and that is why the bioavailability of the drug like paclitaxel is very less when it is given to the patients. There are many alternative approaches which have been done in the pharma industry for increasing the bioavailability, and all these, like by using different types of co-solvent, emulsion, liposome, psychodextrins, nanoparticles, microspheres, the chemical methods like analogs, prodrugs, and the oral administration all have not given many of the successes for the bioavailability enhancement of Taxol. When we have taken this drug for the bioavailability enhancer activity along with piperine, we got wonderful results. This is the type of specific graph which shows that the mean plasma time concentration of paclitaxel with piperine when given by oral routes, the paclitaxel and piperine has shown a shoot up of the bioavailability than that of the conventional dose of the paclitaxel. It indicates that the paclitaxel and piperine in the half dose of paclitaxel increase the bioavailability of this anti-cancer drug. So it indicates that the drugs like paclitaxel or even vinblastine, vincristine, and many of the other anti-cancer drugs, these drugs also are having the type of degradation and metabolism in the liver by cytochrome P enzyme, and all these drugs have been worked out well with piperine, and it has given a very good results by reducing the dose. As it is done with rifampicin, 450 milligram to 200 milligram shift of the dose, just like that in almost all of the anti-cancer drugs and anti-HIV drug, this is a real possibility that the dose can be reduced and economics of the treatments can be changed. The another drugs, in summary, the research of the paclitaxel with piperine indicates that twofold increase of the bioavailability has taken place in paclitaxel, and mostly it increased the T-half, Cmax, and the AUC of this drug when compared to the conventional dose and the dose of paclitaxel and piperine. The another drug which we have taken for this study was atazanavir, which is anti-HIV protease inhibitors. This is the protease inhibitor, which is a very, very good anti-HIV drug. But this drug is mostly, although it is a protease inhibitor, it is mostly broken down in the liver by cytochrome P enzymes. And because of that, this drug is always given along with ritonavir, which is another protease inhibitors and anti-HIV agents. So atazanavir has been, which is a synthetic drug, mostly it is it completely inhibits the viral protease enzymes, but it has to be given along with the another protease inhibitor, ritonavir. And what is the action of ritonavir? Ritonavir is the inhibitors of cytochrome P enzymes in the liver. But because of this addition of ritonavir in a very large proportion, that is 300 mg, mostly ritonavir has its own side effects which the patient has to tolerate. The atazanavir has its own side effects, but along with that, when ritonavir is given, it has also its side effects. So both of the side effects intensifies the ends of the health of the patients. And we thought that instead of ritonavir, if we add piperine to atazanavir, what would be the effect of this drug? So we have studied atazanavir and ritonavir Both of these drugs were studied along with these types of animals. But in these studies, we have utilized piperine also and the another component of tricatu, that is ginger oleoresin also we have studied. That what is the effect of both piperine, ginger oleoresin individually as well as in mixture in atazanavir, and we have got the wonderful results which has reduced the dose of the atazanavir as it has done for paclitaxel as well as rifampicin. So here the graphical representation is there that atazanavir conventional dose, that is 6.5 mg in blue, the atazanavir conventional dose which is reduced to 3.25, atazanavir 6.5 mg with piperine, and atazanavir 3.25 mg with piperine. As we are dealing with piperine, I have not shown the effects of the ginger over here. So it indicates that the effect of the piperine along with 7.5 milligram of piperine along with atazanavir 6.5 mg, how bioavailability has increased in the case of atazanavir. This type of study has been done along with the atazanavir ritonavir combinations also, and in this case also we have seen that the yellow line, which is atazanavir ritonavir combination with piperine, we have seen that the bioavailability is very much compatible with that of the atazanavir ritonavir conventional dose. So this indicates that piperine, which is bioavailability enhancer, which is a very old molecule of about 300 years, 3000 years in therapeutics in Siddha system of medicines. This is the old molecule which has been explored to have the new potential of the bioavailability enhancement when we are studying with novel drug delivery system. I have worked with many of the other natural products like lupeol, boswellic acid, anti-TB, anti-diabetic drug from the fenugreek seeds, and in almost all these cases we have found that piperine increases the bioavailability of all these natural products very significantly. I am thankful to my students like Meha Motiwala, Prachi Shukla, Garima Jain, Swati Prakash, those who worked on this project. Mostly Meha Motiwala worked on anti-TB drug, paclitaxel, and the other M.Pharm students at the College of Pharmacy in Department of Pharmacy, Guru Ghasidas University, they have worked. We have studied with collaboration with Debasis Bandopadhyay, Department of Physiology, Oxidative Stress and Free Radical Biology Laboratory, University of Kolkata, as well as with the Industry for Bio Laboratories, Nagpur. And these were some of the papers published through our research. I would like to take your attention to these slides that when piperine's first research papers came into publications, after that mostly American institutions and the industries, they got activated and they first of all filed the patent on the piperine's bioavailability enhancement. So although Regional Research Laboratory, Jammu, worked for the bioavailability enhancement activity and they are pioneer in this research work, the patents were filed by the peoples, those who have not worked on these lines. And after that, many of the patents were filed by a variety of industries and they have protected their rights, patent rights for almost all types of therapeutically active drugs. So you will not find any of the drug now which has not been worked out with bioavailability enhancer piperine. But it is a very disheartening fact that Although most of the pharma industries, they have filed the patents and they have rights of piperine as a bioavailability enhancer with therapeutically active agents. Except Cadila Laboratories Limited and example of rifampicin, no industry as yet came forward to reduce the dose of the drug by adding piperine and bringing it to the market. So this is a type of research now which requires much more attention, and it should be brought to the limelight so that the pharma industry would reduce the dose of their drugs by using the bioavailability enhancer like piperine or any other types of agents, also bioenhancers. And with that, if the dose is reduced, the side effects of the drug may also be reduced, cost of the drug may also be reduced, and with that, the economics of the total treatment of the patients may be reduced. So with this, I would like to put an end to this. I'm thankful to the organizer of IFS College of Pharmacy, my friend Dr. Professor Gupta sir, and my student. I'm very proud that our student Dr. Sant is working there. Some other students of GGV also are working in IFS College of Pharmacy, and they're doing very well. I'm thankful to all the participants which are here to share this presentation. Thank you very much.
Thanks, sir, for your inspirational word, and thanks for excellent talk on piperine as a bioenhancer for the different type of drugs. So I request to director sir, please speak few words.
Thank you very much, Sant Kumar Verma ji. So, first of all, I am thankful Dr. Vinod Kumar Rangari ji. And the title and topic selected, I am just only focus on the one line. This is the old to new. Old molecule, it can be work as a with the new potential. So this is the need of the potential of the individual scientist. It is not essential that you can always generate a new molecule, but how much work you can do on the old molecule so that it will be the highly effective and useful to the society. So nowadays the most of the peoples, they are working on the application. As you know, our worthy prime minister, he is highly focused only one special thing, and this is the special thing, that how we can utilize our resources and how we can work on the local